Thank you. Thank you so much for this warm introduction. Good morning, everyone. I'm quite sleepy too. So this is the outline of this presentation. So you can see in the title, I had like, no, doesn't work maybe. Okay, that's fine. So we have this word discovery in the title, but actually what we will talk about is also discovery and development, mainly because MR and MR PET can play, especially MR can play mostly a role in the second part, the development. So let's get into it. For those of you, if any of you is not familiar with PET, what we have here is that we inject a radioactive compound into the patient, which is linked to a targeting molecule, pretty much similar to what happens in contrast enhanced MRI, actually. Just that this is a radioactive compound that emits some radiation that it's then that is then detected by gamma cameras. Okay, so we have these gamma rays that can tell us where the radioactive compound is accumulating because of the target substance. So it's very important that we know what we're looking for in the brain, and this keep that in mind for later. So this is an overview of different MR PET tracers that can be used in neurodegeneration. So I would mostly, if not completely, talk about neuro, because this is my thing. You can see that dependently on which tracer we use, we can assess different functions or dysfunctions of the brain, and also that each tracer accumulates quite differently also in the case of healthy subjects, or the healthy subjects that can go through PET. I'll give you the example of F-DOPA, which is used to assess striatal depletion of dopamine, which happens in Parkinson's. So here you will see that this image will only captate the striatum, the subcortical structures, while other tracers accumulate also in the cortex. And then we can look at eventually hypometabolism or hypermetabolism in certain region. And this is done by calculating the specific uptake value ratio. So not only we must quantify the uptake of the tracer voxel by voxel, but we also need a reference region, because actually it's not a quantitative measurement that can be compared across subject. I might have, on average, an higher uptake than another person. Also, older people might have, on average, a lower uptake compared to younger people, and so on and so forth. So why hybrid imaging? It doesn't really display correctly. Anyway, of course, because we can simultaneously study multiple properties of the district of interest, and especially for districts other than neuro, hybrid imaging allows for a better co-registration of the modalities. You may as well, in neuro, you may as well do a nuclear medicine exam and then co-register to a structural MRI and get acceptable results. But in other districts affected by body motion, by physiological motion, this is quite harder. But we will see that PET-MR actually allows a better motion correction. But hybrid imaging is not only PET-MR. Of course, it all started with PET and SPECT combined with CT. And nowadays, I learned this when I started to study about PET-MR. Nowadays, almost all PET systems are actually PET-CT. So it's not that thing. This is why nuclear medicine doctors are not that impressed by PET-MR, because they say, well, we have PET-CT. We don't need PET-MR. So actually, the first images on a human prototype were acquired in 2006. Sorry, but still, the potential of this kind of machines has not been exploited at all. This is like the possible design that could have been developed, especially like the coplanar is typical of PET-TAC, while the integrated is what characterizes most of the PET-MR systems that are available on the market. And this allows us to acquire simultaneously without moving the patient, because both detectors can coexist in the same ring, if you want to say like that. So why PET-MR? Because of course, compared to CT, we have less dose. We just have the dose from the PET. We might exploit the multiple contrast obtainable with MRI. As I said, minimization of axial displacement, better motion correction, because while I'm acquiring the PET, I can acquire an fMRI sequence and correct for motion the PET over the dynamic range of acquisition. I can combine the spatial resolution of MRI to the picomolar sensitivity of PET, and this allows to correctly characterize quantification, the uptake in smaller regions rather than macroscopic structures. And then nowadays, maybe once one of the caveats might have been the longer acquisition times of MRI compared to CT. Now, if we have long acquisition times, usually we also have a lot of sequences acquired. So it's a trade-off between multimodality, multi-contrast, and acquisition time. But actually, if we just want to acquire an MP-ray, a T1 weighted, and the flare, other than the PET, this can be done quite easily. Why not? Please stay with me and don't be scared by all these things. The point is basically PET and MR disturb each other due to their very intrinsic characteristics. And as you can imagine, the bare existence of the PET ring inside the MR scanner is a source of noise and dishomogeneity. And I can tell you, if you don't have a very good coil, your MR images in the PET MR scanner won't be top, let's say like that. The neuroradiologists will have their word on it. But the point is that the coils, the acquisition coils, must be PET transparent to guarantee the simultaneous acquisition of both modality. Okay, so these are all the caveats, and I won't really concentrate on that. But now here, attenuation correction strategies, think about it as like a correction regarding the uptake value, a bit of correction regarding the scattering, the possible scattering, some kind of smoothing of the PET signal, let's say. Most of these things have actually been solved. The main problem, I keep pushing the wrong buttons, the main problem remains the mutual interference of the two modalities. But look at the benefits. So if we are able to properly design an MR protocol according to which is our research question and what we really want to investigate in a specific pathology or in physiology, we would be able to study a lot of different complementary properties. And dependently on the kind of tracer that we use, we can also estimate, I don't know, I'll give you an example. I could estimate metabolism with FDG, fluorodeoxyglucose. It's the most used tracer. It just estimates the glucose consumption at the level of the cortex, or of the cortex and of the subcortical structures. If I acquire an optimized functional MRI that maybe allows me to acquire, to estimate also a PCASL and measures of cerebrovascular reactivity or mitochondrial function, I could extract a lot of information and correlate the metabolism measured by FDG and the cerebrovascular activity measured by optimized fMRI, calibrated fMRI methods. And in an ideal world, what if I find correlates of metabolism that can be achieved with MR, and someday the patient must not go underneath PET and must not be injected with radioactive tracer, but maybe he can go through an MR protocol, because you can use this machine as either a PET or only an MR. You can still use it as a single modality. Well, the challenge is, yes, quantification is suboptimal compared to the one performed with PET-CT, but not so much yet. And the workflow, so it's not that straightforward, but this happens also with multi-contrast MR. It's not straightforward to fuse the two imaging modalities when we try to do advanced post-processing methods. You have to do it on your own, let's say. There are not that much softwares available. So let's get back to theranostics. We have heard a lot of times that it's the crisis of therapy and diagnosis. So we diagnose the disease, but we also monitor drug efficacy over time. So, and we have also heard that this is a very long, from discovery, pre-discovery, to development and then approval for clinical practice. There's a long road and a lot of money that needs to be gained and then spent to develop these things. A lot of challenges even here, though. In the case of neurological disorders, the key, as I showed you before, the key thing is you have to know what you're looking for in the brain. You're looking for abnormal accumulation of a protein. You're looking for depletion, for the death of some specific neurons, and so the lack of neurotransmitter of the substance those neurons produce. But the point is, many neurological disorders do not have a clear pathophysiological basis, or many of them share pathophysiological mechanisms. Think at the dementias, the different form of dementias. Some of them overlap with movement disorders. So you have like a spectrum of diseases that might have the same PET abnormalities, depending on the tracer. And then so I don't know what I have to cure, basically. I don't know where my drug needs to act. I'm not sure of what is the target of my drug. Then the drug itself must be with a low molecular weight, must be radiolabelable with this specific tracer. So we also have to find, once we know what we want to seek, we need to know what actually binds to this molecule. High affinity, so the tracer has to have an high affinity for the target of interest and a low binding to other regions. Because then I have to normalize. I have to know that it won't just spread and distribute over the district of interest. Blood-brain barrier permeability. This is, I probably should have put it in the front. The BBB is there to block substances from entering the brain. So actually, this is why nanomolecules and other things are being studied. And of course, anything that we put inside our body and it's not natural might provoke some toxicity. So I'll show you now a few examples. Tell me if the time is running out. Huntington disease. So Huntington disease is one of the few, it's a very severe neurological disorder that inevitably leads to the death of the patient, to a very bad death of the patient. The good news is we know why Huntington develops. 5 minutes, okay. We know why Huntington develops. There's a gene that codes for a protein that generates abnormal repetition of some triplets. The point is, these Huntington, HTT, these mutated Huntington fibrils accumulate all over the brain, starting from the subcortical structures, leading to a widespread degeneration of the patient, widespread and quick. So, as I said, there's a CAG repeats in this chromosome that we might have. We have these repeats, but if the number of these repeats goes over 39, says this article, I knew 36, but we're there. The more triplets we have, the more severe the disease will be, and the earlier the onset of the disease will be. Luckily, this is rare, but the point is, can we quantify mutated Huntington with PET? And eventually, can we develop a drug that can maybe destroy these fibrils and these tangles? Well, not yet, but there's been a lot of research and also future challenges ahead regarding the clinical tracers that bind to mutated Huntington. These are long story short, we are down to fluorine and carbon tracers. And there's, of course, there are a lot of medications that are also being studied to reduce the load of mutated huntingtin fibrils, and possibly these tracers will reach a specificity so that we can try to monitor the disease. So these are patients, in a very cynical way, it's the only hope they have, actually, to get some medication. This is here again because, maybe there's no, none of the tracers, but basically we want to see huntingtin. You would see something like this, because the other thing is this mechanism actually results in an abnormal function of cholinergic system, GABAergic system, cannabinoid receptors, glucose receptors. So there's also a wide variety of pathophysiological processes, and then sensitivity and specificity of the tracer become like a real struggle. Let's see the other case, most common. A lot of research has been done on this. So in Alzheimer's disease, we know that we can look for both two types of protein misfolding. There's amyloid beta plaques, and there is tau tangles. These are two proteins that accumulate in Alzheimer's. But what researchers have found over the years is that amyloid beta might be present, plaques of amyloid beta might be present also in healthy controls, in cognitively spared healthy individuals. While tau is probably more specific of the pathology. So the choice is, what kind of drug do I develop? Do I target amyloid beta plaques or do I target tau pathology? I'm almost at the end. And there's also here a lot of tracers. There has been a lot, as you know, a lot of research on Alzheimer's disease. And so we can look for hypometabolism, FDG PET, and amyloid PET tracers. These are the molecules. I won't dare going into deep of the chemistry below. We have tau PET tracers for the diagnosis. So these are currently being used, tau more than beta amyloid, to select patients that are then included in clinical trials for new medications. And then the load, the uptake, the SUVR of SUVR of these tracers is used as an endpoint of the clinical trials. So this is a kind of the first maybe theranostic approach that we've seen in neurology and neuroimaging. But as I was saying before, now small molecules treatments are being, the process is always the same. We try to look for a bigger molecule that must embed some substance that then is delivered where it has to be delivered, where it needs to act. So to sum up, these are my personal considerations. PET MRI is underexplored currently, also both in clinical practice and research settings. It might not ever be a clinical tool, actually, but it could be very useful, a much more useful tool in research too. And also the theranostic approach, so the use of imaging techniques to deliver the medication directly into the brain is probably the way to go to try to find a cure to neurological disorders, because no neurological disorder actually has a cure, not even Parkinson. We can cure some symptoms, but we cannot stop degeneration. Promising drugs are being developed, but there's so many prerequisites they have to meet to be even tested in preclinical settings that it's very difficult that they arrive at. We've seen it a lot. It's very difficult to see them arriving to a human clinical trial phase. And I think this is it. Thanks. Thank you for your attention.